Hello, it's wonderful to be here again. I always think one of the most nerve-wracking things in my entire science career is introducing speakers for when they give talks. I don't know why it's so frightening. It gets even more frightening when it's your PhD advisor. You go straight back to the feeling you had when you were first meeting with that person in office 20 years ago. You feel the same. But it's also a real privilege and an honor to introduce our keynote speaker, Linda Partridge. Also, it's easier when someone is famous enough they are both knighted and have a Wikipedia page. You can go and check what their CV is on Wikipedia. So if I get anything wrong here, you can go to Wikipedia and check out the real information. And I want to give you a few facts about Linda from her CV that are mainly from Wikipedia, which I didn't fact check, but we'll check. So Linda Partridge is currently the Weldon Professor of Biometry at the Institute of Healthy Aging and the Department of Genetics at University College London. She was also, of course, it's been there for many years, but also, whilst doing that, moved to Cologne and kind of were two people at the same time to become one of the inaugural directors of the Max Planck Institute for Aging, where she's now emeritus, and set up that wonderful institute, which many of you will know about. She's got a long list of awards, and I checked them and found that most of them came after I'd left the lab, which means that you can either choose whether I was responsible or holding her back. I don't know. I'm going to go for one of them, but I read some of them. So some came before. So Linda was elected as a Fellow of the Royal Society in 1996. She was also, you might not all know the various levels of royal decree you can get in the UK. If you don't, you can Google it. But she was appointed Commander of the Order of the British Empire in 2003, which is a very big deal. Not being done with meeting the Queen, I think at that point she went back again to meet now who's the King in 2009 when she was appointed Dame Commander of the Order of the British Empire, which is a very fancy way of saying she was knighted, which is an incredible thing for a scientist. She was elected to the Academy of Medical Sciences in 2004 and awarded the Linnean Society Darwin Wallace Medal in 2008. She was also, she's a foreign honorary member of the American Academy of Arts and Sciences in 2010 and the German National Academy of Sciences in 2023. And Linda's career has spanned just so many different areas, from evolutionary biology to fitness trade-offs to genetics to software aging to mouse aging and all these different things. And she'll talk to you about many of those tonight. I think a little bit of how geroscience field has led, really how she came into the geroscience field and led to what she's done now. She's also just been a wonderful mentor to many, both in her lab and I think in the field, and has really been pivotal in many people's careers, including my own. So it's a real privilege to welcome her tonight.
Thank you very much, Will, for an embarrassingly kind introduction. It's a real honour to be giving this first talk in this famous auditorium at Cold Spring Harbour. So thank you very much to both of you and the sadly absent Meng for inviting me. Really nice. And you've put together a fantastic scientific programme for this meeting. I think there's something going wrong with the sound. Can you hear me? Yeah. I think you've put together an absolutely brilliant scientific programme for the meeting, and I'm sure we're all looking forward to what's to come. So I'm going to talk about geroscience and particularly repurposing of drugs for geroprotection. So the basic idea here is that because the various mechanisms of aging are acting as the major risk factor for aging-related diseases, then perhaps we can intervene in these underlying mechanisms to prevent or delay the onset of diseases, and not just one disease. So the key points here are prevention and the one-into-many, single intervention, but protection across a reasonably broad spectrum of disease. And over the last 20 years or so, I think we've come to a really quite mature understanding of the underlying mechanisms of aging. They've been characterized in various ways, including with these hallmarks of aging, which specify the things that go wrong at the level of the genetic material itself, the way it's packaged, the way it's expressed, the way that things are controlled in cells and cells communicate with each other. All of these things are going wrong in parallel during aging, and they're also interacting with each other. They're not independent. It's a complicated process. The contribution of these different mechanisms of aging differs between different tissues and different kinds of animals, but these are the basic things that go wrong during the aging process. And they've all been shown experimentally, if you interfere with them, to improve some aspect of health during aging. So interfering with them is experimentally demonstrated to be geroprotective. And they're also all present to some extent in the etiology of the things that go wrong in this particular diagram in human aging. So subclinical things like loss of bone mass, gain of abdominal fat, frailty, but also the diseases of aging, and particularly this one, multimorbidity, the presence of more than one disease in a single individual. And that's where we're expecting these geroprotective interventions to be particularly powerful, because they can prevent multiple diseases simultaneously. So this is something that we've been looking at to start with actual human data from patients and from epidemiological cohorts, trying to look at whether there's actually evidence for this point of view that interfering with these mechanisms of aging could prevent patterns of multimorbidity. And this has been a real collaborative effort, mainly at UCL. And the first thing that we did was to simply try to define which diseases are age-related in their instance. So which diseases become more likely to occur for the first time in patients as they get older. And to do this, I worked with these people. So Val Kuhn is the most extraordinary person. She started out as actually a person who did risk analysis for insurance companies. She then ended up on the trading floor of one of the banks, and she got bored with that, so she retrained as a GP, and then she decided to do a PhD, and these are the results of some of her PhD work. So what she did was to consider 278, I think, high-burden diseases, and she worked with the very good national health records in the UK, so primary admissions and also hospital admissions. And she had data for about 3 million patients. And her PhD was supervised by Arun Hinkarani, who's actually a cardiologist, but also interested in analysis of epidemiological data. So these are the diseases she looked at, and the cohort that she looked at them in. And what she's done here is to do a cluster analysis to identify the ones that are age-related in their instance. And they're in the first 4 categories here. So essentially, as you go through those 4 categories, they've got an earlier and earlier age of onset. So the most extreme is cluster 1. Very few diseases there. For instance, dementia is one of those. The 21 in cluster 2 are mainly cardiovascular things. Most cancers are in cluster 3, and a lot of cluster 4 are digestive. But the really bad news here from the point of view of the aging process is that 207 out of these 284 diseases are age-related in their instance. So age really is the very major risk factor here. So what we wanted to know, of course, was whether the hallmarks of aging were acting as risk factors for combinations of these diseases. So for that, we collaborated further. So Helen Fraser actually did this project as her PhD work. Andreas Bayer and Ronja work in Cologne, and they're particularly strong on network analysis, which we needed for this project. And Madeleina's very good at artificial learning and literature mining. So what Helen did was to take the hallmarks of aging, and at the time we did this, we didn't have the second generation, so she worked with the original 9 hallmarks. And what she's done here is to explode them into a dictionary, because if you actually look at how things like mitochondrial dysfunction or DNA damage are referred to in the literature, there are a lot of different terms that are used to describe each of them. So that is what this dictionary is. It's a real list of all the ways that they're actually referred to in the literature. And she then mined almost 2 million abstracts in PubMed to look for co-mentions of the hallmarks with the age-related diseases, the 207 that have been identified by Val Kuhn. And she used something called the Okai coefficient for co-occurrence. What that does is to take into account the number of mentions of each hallmark in each disease. So it's really the co-occurrence that's important. And she then went and actually checked the abstracts manually to make sure that the co-mentions were causal, so that the suggestion was that the hallmark was causal in the etiology of the disease. And what that enabled her to do was to define the top 30 of those 207 diseases that were associated with each of the 9 aging hallmarks. And this is what that looked like. It's the columns on the left. So we've got the 9 hallmarks across the bottom here, and then the various diseases with which they were associated. And you can see that it's an interesting and quite complicated pattern. So, for instance, breast cancer, all of the hallmarks are associated with breast cancer, whereas some are much more specific. So, for instance, further down, low HDL is associated only with disordered nutrient sensing. So the interesting and complicated patterns there. But what came out were many of the things that you'd expect, listed on the right here. So genomic instability and epigenetic alterations associated with primary malignancies, proteostasis neurodegenerative disease, nutrient-sensing metabolic disease, and so on. So a lot of what you'd expect came out. And as I said, the Okai coefficient actually controls for differences in study intensity of different hallmarks and different diseases. But nonetheless, once a co-mention happens in the literature, then other scientists may be more likely to study that association. So to try to look further at whether this is real biology or just, to some extent, the behaviour of scientists, what Helen did was to take the top 30 diseases that were associated with a particular hallmark. She then went and looked at the GWAS catalogue, the Genome-Wide Association catalogue, to see if genes had been identified as associated with as risk factors for that disease in genetic analysis. And she could then look at what those genes were annotated for. And what she found was that they were very heavily annotated for the hallmark. This is a huge data set, actually, but this is just 2 of the examples. So, for instance, for stem cell exhaustion on the left, if she takes the annotation of the genes associated with the diseases that are associated with stem cell exhaustion, then this is what she gets, and it's all stem cell annotation. Similarly, for deregulated nutrient sensing on the right. So it looks as though these associations are real biology rather than anything to do with biases in the literature. But the important question, of course, is can this association with different aging hallmarks actually explain the co-occurrence of the diseases in patients? And to do that, to look at that, she did a network analysis. So she took all of the 83 diseases that were associated with aging hallmarks, and she made an overall network of their co-occurrence in the patients, in Val Kuen's patient population. And she could also do that individually for each aging hallmark, which is what she's done here. So it's the 83 age-related diseases. She looked at 4 decadal networks starting at 50, so 50 to 60, 60 to 70-year-olds, and so on. And she made a subnetwork of 30 diseases that were associated with each hallmark. So just as an example, this is the one for mitochondrial dysfunction in patients aged 50 to 59. These are the 30 diseases, so the darker the colour, the stronger the association of the disease with the hallmark. So there are a lot of cancers down here in the left, other diseases on the right. And what she could then ask was, is the association between the diseases in these subnetworks stronger than in the overall network for the 83 diseases? And she could ask that for each of the 4 decadal intervals in the patients. And it turned out that they were. So for nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication, there were stronger associations in patients with the diseases associated with that hallmark than you would expect at random from the associations. And quite remarkably, they were stable over those 4 decadal intervals. So it really does look as though the aging hallmarks have explanatory value for disease associations in real patients. We don't know why those 5 and not the other 4, or to some extent what's driving the associations. But obviously one possible explanation is different in lifestyle between different patients, exaggerating the progression of the aging of a particular hallmarks and then the diseases that are associated with it. So that's something that we've tried to look at more recently, but this time just using standard demographic data, not patient data, from cohorts that are well characterized during aging. And this has very much been the work of Mika Kivimaki, who's actually a colleague of mine at UCL. And what he's done here is to take data from initially healthy individuals from two different populations. One's the UK Biobank, which is a fantastic cohort source of information, and the other is a Finnish cohort that he frequently works with. And what he's done is to look over the ensuing period about at whether diseases associated with aging hallmarks are likely to co-occur in particular individuals that have specific risk factors. So to make that a bit more concrete, over here on the right, we've got the UK Biobank at the top, the Finnish cohorts at the bottom, again, the 9 aging hallmarks, because he's working with the diseases that were identified in Helen's study. And he's got these risk factors, so obesity, current smoking, education, depression, and so on. And what he's asked is, are 3 or more of the diseases associated with each of those hallmarks more likely to occur in patients over the next 10 or 20 years if they're obese, current smokers, and so on. And you can see the hazard ratios here, and they're coloured up. And what you can see is that obesity is the major risk factor for a lot of the co-occurrence of these hallmark-associated diseases, but also current smoking and education play quite a role. And there's a slight difference between the cohorts, which is probably to do with the demography of the cohorts, actually, than real differences in biology. But I think what we're starting to see here is risk factors associated with hallmarks and explaining these disease associations. So obviously there's a lot more work to do here. It'd be nice to work with other cohorts to make sure that the original findings from the Fraser study hold up. And also it would be nice to work with the current hallmark list. But I think these are really saying that what we're expecting in humans is that if we tackle these underlying hallmarks and the risk factors for them, then we should be able to prevent multimorbidity. So, how can we intervene to prevent multimorbidity? We have on the left the main interventions that people are using into the hallmarks. Genetics is used mainly for discovery. And of course, as the Kivimaki study suggests, lifestyle factors are still one of our best interventions. So diet and exercise are still very powerful. But even people with a blameless existence nonetheless get the diseases of aging, and anyway, compliance with lifestyle factors is not high. So we would like to be able to develop other interventions that could help prevent multimorbidity. And the ones that we've been particularly working on are the nutrient sensing network, this dysregulated nutrient sensing, and drugs. So as I'm sure you all know, when we're dealing with the nutrient sensing network, this is essentially a good times signaling network. If nutrients are plentiful, stresses are absent, then it signals with strong intensity, whereas if stresses are present, then it activates various transcription factors that induce stress resistance. And a huge amount of the work on this has been done with these model organisms. They've been real engines of discovery, and they're still engines of discovery of mechanism. And of course, the hero here is the worm, and Cynthia is sitting in the second row of the audience. It was really here that the importance of this nutrient sensing network in aging came clearly to light. And it's since proved to be really heavily evolutionarily conserved between the different model organisms, and also genetic association studies in humans are starting to implicate some of the transcription factors, particularly that are important in this network in human aging. So what are we actually talking about here? This is a highly simplified, even by Drosophila standards, version of the network. So what we're talking about is extracellular ligands, so insulin, IGF. There are actually 7 of them in Drosophila. Receptor tyrosine kinases, just a single one in the worm and in the fly. Insulin receptor substrate called Chico in the fly, signals just below the receptor. And then the bifurcation of the pathway into the famous canonical pathway made famous by the discovery of the role of FOXO in the worm. So if you remove that transcription factor, then reduction of upstream signaling of the pathway no longer extends the lifespan of the worm, and the same is actually true in Drosophila. But also sitting in plain view, although largely ignored for a long time, is the RAS pathway, very important in cancer, which signals in the fly to anterior open, but generally to E26, transcription factors. And it's clear from the genetic evidence in the model organisms that suppressing the activity of this network is beneficial to health during aging. It extends lifespan and improves function during aging. And we've become particularly interested in two drugs that disrupt its function and are therefore potentially geroprotective in humans. So these are already licensed drugs. So the first is well known to many of us, rapamycin, which disrupts the mTORC1 complex. This is a very ancient, evolutionarily conserved intracellular bit of the signaling network that particularly senses amino acids but also stresses. Rapamycin's on-label use is a cancer chemotherapeutic to prevent rejection of organisms after transplant and to prevent restenosis after cardiac surgery. We're also interested in trametinib, which inhibits MEK kinases in the RAS signaling pathway. So why these two drugs? We've already got very strong evidence from the animal studies that genetic disruption of mTORC1 can be highly beneficial to health during aging. And a lot of the work that we've been doing is trying to understand whether actually intervening at the level of mTORC1, or indeed of MEK, is the best level at which to intervene in the network. What we want is this one into many, so a disruption of a single protein or protein complex, which produces the maximum beneficial effect on diseases during aging, preferably with the fewest side effects. So there's an issue with this network, which is very complicated. There's a lot of feedback, has a lot more components than are shown here, of identifying the right place to go in. So we're very interested in the mechanisms of what these two drugs are doing. Rapamycin works well in flies, discovered a long time ago by Ivana. You can see they're not huge extensions of lifespan with the drug, but it definitely extends lifespan. And one of the benefits of doing this kind of drug work in invertebrates is that you can do epistasis with genetics relatively quickly, and that enables you to get down into mechanisms. And when Ivana started to do that, she found that it was entirely inhibition of the TORC1 complex that's important. TORC kinase is present in two complexes, and TORC2 is to some extent inhibited by rapamycin, but that's irrelevant to this aging story in Drosophila. And she could show by blocking them that increased autophagy and suppression of the S6 kinase were required for the extension of lifespan. If she prevented either of those things happening genetically, then the drug no longer extended the lifespan of the fly. And she also found that the lifespan extension is largely confined to females. And this has been picked up subsequently by two postdocs in the lab, Jenny Reagan and Yuxuan Liu, both of whom have their own labs now, to look very thoroughly at this sex-specific effect. So you can see the basic effect on the left here at the dose we usually use. There's no extension in males, quite a decent one in females. Males, we give the drug to the flies in the food, and males eat a lot less than females do. So they wanted to be absolutely sure that even if they upped the dose, they didn't see anything in males. And indeed, you can see on the right here that even at doses that are starting to shorten female lifespan, we see nothing in males. And we know the drug's getting into males. We see the suppression of the S6 kinase activity and so on. It's not that the drug isn't working. It's just that it doesn't extend their lifespan. And in Drosophila, that's a really quite strong hint that it's probably something to do with the gut, because there's a very big difference in the way that the gut ages in the two sexes. So you can see at the bottom here, males. What you're looking at is a columnar epithelium, very like a small intestine, a nice row of nuclei on the basement membrane. And as the males get older, basically nothing happens. It's fine. And if you look down on the surface of the tips of these cells, again, you maintain the nice honeycomb arrangement. But in the females, what you see, even in quite young females, is that the stem cells that are present in the gut start to proliferate. You get dysplasia, little misdifferentiated groups of cells at the outer edge of the intestine. It pulls away from the basement membrane. You start to see wound-healing rosettes. And eventually, it becomes completely disordered. And in quite a lot of the females, it becomes leaky. So there's a real gut pathology during aging. And that's rescued to a large extent by rapamycin. So we've got the proliferation of the stem cells here. You can see that the basal level's much lower in males and it's unaffected by the drug, but it's reduced from its very high level in females. These are these little dysplasic groups of cells, and you can see that the drug basically takes that down to male levels in the females. This is gut leakiness, which is measured with a blue dye that leaks out of the gut into the fly. And again, you can see even at basal levels, it's much lower. It hardly happens in males. About 20% of females show it, but it disappears if you give them the drug. So it really rescues. And it turns out here that the reason is the autophagy part of the story. It's this induction of autophagy. So there's a very big basal difference in the rate of autophagy in the two sexes. So here we've got lysotracker staining. This is quantified over here in females and males. And what you can see is if you look in the absence of the drug, the males have a much higher lysotracker staining in their guts than females do. And if you give the female rapamycin, it brings her lysotracker staining up to male levels. It kind of abolishes the sex difference. The point of the cyto-ID here is to distinguish between blocked autophagic flux and increased autophagic flux. And what you're expecting, if it's unaffected by the drug, is that it's an indication that the flux is increased. So what we're seeing here is that males have higher autophagic flux to start with, and females get to male levels if you give them rapamycin. And one of the nice things about working with flies is that the sex determination is cell autonomous. So each cell in the fly decides what sex it's going to be in accordance with its karyotype. There are no sex hormones. And the mechanics of that sex determination cascade are very well understood. So you can actually transdetermine individual cell types in the fly by taking advantage of that to misexpress the sex determinants. And if you do that with the big absorptive enterocytes in the gut, which is where most of this lysotracker signal is coming from, then what you find is that the autophagy goes with the sex of the enterocyte, not of the fly. So here, just to give you one of the examples, you've got the converse over here. We've got males, and we've seen that they have high lysotracker staining in both the presence and the absence of the gut. If you give them female enterocytes, they now have low basal lysotracker, which increases when you give them the drug. And the converse happens over here. So this autophagy phenotype and its response to rapamycin is going entirely with the sex of the enterocytes. And in those very same flies, if you look at their lifespan, the same thing's happening. So here on the left, we've got, again, males and males with female enterocytes. In blue, the males, as you've already seen, don't respond in their lifespan to rapamycin, but if you give them female enterocytes, they now do respond, whereas over here, females respond to rapamycin, but if you give them male enterocytes, nothing happens. So it's going just with the sex of these cells in the gut, which implies very strongly that the autophagy is causal. And not to drown you in data, we can show that in a variety of ways. It's easy to genetically directly induce autophagy in gut enterocytes. And if you do that, you prevent the gut pathology in females and extend their lifespan. But the main point I'd like to make about this autophagy story in the gut and the one into many is there's actually a bewildering parallel array of mechanisms going on here, even in a single cell type in the gut. So when we give them the drug, these three things happen in the enterocytes. It induces autophagy. There's canonical induction just through ATG1, standard macroautophagy induction. There's also a completely non-canonical process. So if you look at the guts of these flies that have fed the drug, there's a huge increase in expression of histones. There's increased nucleosome occupancy. There are changes in mRNA expression, especially of a particular autophagy cargo adapter called blue cheese in the fly. It has an exact mammalian ortholog. And this is the basis of the sex difference. So it's actually this non-canonical. So males have a higher expression of histones and all the other things than females do. It's not the standard induction. But there's also a massive memory effect of this drug. So if you give adult female flies rapamycin for, say, the first 10 days of adult life and then take it away, or if you induce autophagy directly through ATG1 with an inducer for 10 days and then take the inducer away, and then wait, what you see is the females don't get gut pathology as they get older, and they show the full extension of lifespan that they have with chronic rapamycin treatment or chronic induction of ATG1. So it's a permanent 100% memory effect, and that's produced by this positive feedback mechanism. Basically, autophagy induces itself, and it stays up, and it's because of expression of these alpha-monosidases and lysozyme. There's a positive feedback loop that goes on here. So even in this single cell class, we've got these parallel mechanisms, and we don't really understand whether they're acting in exactly the same way, whether it's the same carriers, the same reasons that gut health is being improved. We really need cell biology here to understand what's going on. But there's also a further mechanism just in the gut. This was shown in Nazif Alik's lab some years ago, that also it acts in intestinal cells to increase gut health. So it's these stem cells here that give rise to these endocrine cells and the enterocytes that I've already mentioned. And here it's through repression of the activity of RNA polymerase 3. So there's an inhibitor of POL3, which is called MAF1, and it's directly phosphorylated by TORC1. And that's the mechanism that's operating here. Why that improves gut health is not clear, but it is clear that that's the molecular mechanism that's going on. So just in this single tissue, we've got this multiplicity of mechanisms. And many of them are reflected in mice. So for instance, if you give mice rapamycin, you see the increase in histone expression. You see the expression of the ortholog of the blue cheese that we see in Drosophila. If we look at some of the histology in the gut, so in mice that are now 12 months old that started to get rapamycin at 3 months, if you look at the tight junctions between the columnar epithelial cells, what you find is that they decline with age, but that's rescued by rapamycin. You can look at a measure of gut leakiness, which is this lipopolysaccharide binding protein, which you find in circulation and is thought to be an indicator of bacterial leakage from the gut. That increases with age, but again, it's rescued by rapamycin. And particularly if you look at the critical Paneth cells, which are the neath for the stem cells, they have these granules which really deteriorate during aging. So you can see here the packing of lysozyme in these cells becomes really diffuse. That's rescued by rapamycin. You also see crinophagic and diffuse granules, and again, that's rescued. So mice show really quite early age-related pathology in the gut, and again, it's rescued. So there seems to be conservation there. But you remember I mentioned we also need to see suppression of the X6 kinase, and it took us a very long time to figure out what was going on there. And I don't want to steal anyone's thunder. So Pingxi Yang, who was a graduate student in the labs, now a postdoc at Yale, was the one who helped really to crack this one. This is the title of his talk. He's talking tomorrow morning on Wednesday. But just to make the point about this one into many, in a nutshell, this is what they found. So it was James who actually figured out that it's acting in the white the fly equivalent of mammalian liver and white adipose tissue, and it's basically an inflammation and immunity story. So it increases the expression of the syntaxin 13, and the important thing here is the endolysosomal system, which takes molecules, surface receptors such as this one, to the cell membrane. And it turns out that this is being regulated by the S6 kinase in the fat body of the fly. And what you eventually see by a process that Pingxi will describe is prevention of inflammation and immunosenescence. So this is a different tissue and a completely different set of mechanisms from this one drug. And these are just the ones that we know about. There may be others that we haven't yet found. Coming on to trametinib, the reason this one came to light was the work of these two, who again both now have their own labs. I've already mentioned some work from Nazif's. And they were interested in the role of the RAS pathway at the time, specifically in lifespan extension by loss of Chico. So if you knock out the single fly insulin receptor substrate, you get a long-lived fly. And we've been able to show that that requires FOXO. But what these two did was to engineer the insulin receptor substrate. It's a very conserved molecule, and the way in which it communicates with these two pathways is well understood. So what they did was to mutate the sites, either that communicated with the canonical pathway or that communicated with the RAS pathway. And what they found was that both extended lifespan to the same extent as loss of Chico itself, so they're equally important. And this transcription factor, AOP, was crucial when they did a genetic analysis of this pathway, showing that indeed if you suppress its activity, it can extend lifespan. So it may look like a rather odd setup, but of course what you have here is two pathways that have other inputs that aren't shown here, and they signal to these two transcription factors which actually have overlapping sites where they bind in the nucleus to different genetic sites, but they also have separate ones. So this is a very finely tuned network where you've got these two pathways listening to slightly different inputs and modulating the effects of these two in both overlapping and separate ways. And of course, this discovery really invited drugs, because this pathway is extremely important in cancer, so there are lots of drugs that go for targets. And the one that Kathy identified as really effective was trametinib. So, of course, this is a drug that was developed for human use. It's used to treat malignant melanoma, mainly, but it works beautifully in flies. So these are data from fly S2 cells, and what you can see here is the inhibition of MEC, so looking at phosphorylation of ERK, you see a nice dose-dependent reduction in the presence of insulin, and it's reasonably specific in there's no effect on the phosphorylation of either AKT or the S6 kinase. So the drug seems to work quite nicely in flies, and it also extends their lifespan. So you can see the dose-dependent increase in the purple and red. If you give them too much, the young flies really hate it, but you still see an extension of maximum lifespan. And we're less far with understanding the mechanisms by which this one's extending lifespan, but again, we're seeing the multiple mechanisms story. So the person who's really working on this is Enrique Urena, who's in the lab at UCL. There's a very similar mechanism to the one discovered by Nazif for rapamycin, which is this MAF1 story, again, a repression of RNA polymerase 3. This time it's ERK that directly phosphorylates MAF1 to produce this inhibition of POL3 activity. But it's also acting in the fat body, and again, there seems to be an inflammation story, but it's not the same one as discovered by Pinksy. There's something different happening. So again, it's a pleiotropic drug, and I suspect this is going to be typical of geroprotectors. But because of the very strong conservation of the RAS pathway, we became very interested in whether this drug might be geroprotective in mice. And this is something that we pursued before I left Cologne, and the results are shown here. So Lasonia is the person who's really been leading this. She's now a postdoc in one of the other labs in Cologne. And what you can see here is flies fed 2 doses of the drug throughout life, and females at the top, males at the bottom, and you can see that both doses in both sexes extend lifespan rather clearly in a dose-dependent way in males. We were incredibly cautious with this drug because nobody tried it in mice before for geroprotection, and so we did a dose-response curve for the first 8 weeks, trying to look for potential side effects. And we didn't see any side effects at all with these 2 doses, but nor did we with a higher one. But because we were going to do chronic treatment, we were cautious. We were getting inhibition of ERK with these 2 doses, and so we went with them. But I think we can almost certainly go higher with this drug. But what's clearly the case is that it seems to be geroprotective, and at least at these doses, seemingly without side effects. These are the phenotypes that we actually looked at in the mice. There are a couple at the top. You've seen the lifespan. And there's a decline in heart rate during aging, and that's prevented, and change in respiratory ratio. But mainly, it's a story about inflammation and a little bit about tumours. There are a lot of things it doesn't affect at all at the bottom here, but what we see is that there's an increase in glucose uptake in the brain, which is thought to be to do with age-related brain inflammation, and we see a reduction in that. We see age-related microglial activations down, one plasma inflammatory marker, reduced spleen weight in females. Not so much effect on gene expression in females, but huge downregulation of genes involved in innate immunity and cell division in the male spleen. So there seems to be some sex specificity in the effects here. But clearly what we need to do here is have a harder look at this drug in mice and at a higher dose to really specify these phenotypes. But it's looking interesting. Now, I've mentioned the many targets and also the fact that in the fly, rapamycin does some of the same, but also some different things. And the pathway, the signaling network is a complicated one with a lot of feedback. We therefore became interested in whether combinatorial suppression of different nodes might be beneficial, either because it's stronger or they induce different responses to each other or they prevent feedback in the network. And a long time ago now, Jorge, who's a postdoc in Keith Blackwell's lab at Harvard, looked at this. And we were actually also looking at lithium at the time because it extends lifespan in flies. But here are the data just for rapamycin and trametinib. And you can see the single and double dose response. So these are the two doses of either trametinib or rapamycin that maximize the lifespan extension. You can see that trametinib does a slightly better job. This is what happens in turquoise if you put the two drugs together. You see a completely additive increase in lifespan. And we don't know what the reasons for that are at the moment. We're starting to look at it. But, of course, we were interested in whether it might also be happening in mice. Rapamycin, of course, famously extends lifespan in mice. It was the first drug clearly shown to do so in the ITP, led by Richard Miller. You can see the nice dose-dependent increases in the two sexes here. But it does have side effects, and one of them is metabolic. It makes at the higher doses, the mice become insulin resistant and glucose insensitive. And some work from Dudley Lamming's lab suggests that if you give them the drug intermittently, you can actually rescue those metabolic effects. So we have actually investigated directly intermittent versus chronic treatment with rapamycin. This was Maruf Baghdadi, who's now a postdoc in Cologne. And what you can see here is the effects on lifespan, either of chronic treatment with that highest dose that produces the maximum increase in lifespan, or with 1 week on, 1 week off intermittent treatment in the two sexes. There's actually no statistical difference in the lifespan extension between the two, although I think it looks a bit suspicious. I suspect the intermittent treatment is slightly less effective. But what it does is to rescue the metabolic effects. So technically, at least, it extends the lifespan to the same extent as controls. But what we see is that there's no problem with the metabolic health of the treated mice compared with the controls. However, it fails to completely recapture some of the other benefits of chronic treatment. And I think part of the problem here may be that 1 week on, 1 week off is actually not sufficient to get the rapamycin up to the dose that you see with chronic treatment in circulation. I think it may require slightly longer alternation periods to produce the best effect, but I think somebody else is probably going to have to investigate that. What we have done is to look at the effects of combinatorial drug treatment, because this is what we were really aiming for, and for that we use the doses of trametinib that you've already seen together with the intermittent treatment that you've just seen from Maruf. And this produced really what we thought was quite a striking effect. So you've seen the green data before, and you've seen the red data before. That's the intermittent rapamycin treatment. The orange is what happens when you put them together. And you really get a really quite substantial increase in lifespan in both sexes. The actual numbers are given down here. So it's obviously of considerable interest to understand what's going on here. This is an additive increase again. It's not synergistic, it's not epistatic. They just seem to add on to each other in their effect on lifespan. But what we've done, or what I've done here, is to extract what this double treatment does that neither single treatment does alone, what's going on with this combination. And the story is very strongly one of reduced inflammation and reduced tumorigenesis, probably connected with each other. So we see now reduced tumours in liver and spleen in both sexes, a complete block of this age-related increase in glucose uptake in the brain, blocks the activation of microglia and astrocytes, reduced age-related inflammation in the kidney. Inflammatory markers are down in plasma, especially in females. Again, that seems to be a sex difference. Spleen pathology is down. And genes associated with inflammation and immune response are down in both spleen and muscle. So it really seems to be an anti-inflammatory drug combination. And it harks back in quite an interesting way, I think, to what Helen discovered in that original study where she looked at the association between hallmarks of aging and age-related diseases and their co-occurrence in patients. Because you remember she went on to identify the genes and their annotation that were associated with the diseases that went with each hallmark. But what she found was that there were some annotations that went across all the hallmarks. So the diseases associated with all 9 had annotations in common. And interestingly, 2 of them were reduced activity of the ERK1/2 signaling cascade and innate immunity. So these are just the data for the ERK1/2 cascade, just one way of illustrating these kind of data. So the affected diseases around the bottom here, these are the genes that led to the annotations for those diseases, and the stripes give you the associations between them. So you can see that there's really quite an array of genes associated with the ERK1/2 cascade that go across all of these different age-related diseases. And the same was true for innate immunity. So I think these mouse studies are producing phenotypes that are quite consistent with the sorts of effects that we're seeing in patients. So, to summarise, targeting mechanisms of aging, I think, could delay or prevent multimorbidity in humans. The nutrient sensing networks are a promising target for repurposing of existing drugs. Geroprotective drugs can act through multiple parallel mechanisms. They're very pleiotropic. And pharmacological inhibition of MEK in the RAS signaling cascade can extend mouse lifespan. And we may be looking eventually at targeting this network with a polypill. So, thank you very much for your attention, and I'm happy to take questions.
Amazing talk. So you showed rapamycin can increase both male and female mouse lifespan, but in fly only work on female. Can you do you have any hint about the differences of the species, and also can you speculate the effect on our human? So as a male, should I use the fly data or the mouse data if I consider taking rapamycin?
Well, it's good to get round to the what should I take question right at the beginning. So I think the fly-mouse difference is very much to do with the biology of the 2 species. I mean, flies are extremely sexually dimorphic. The females are machines basically for eating and pumping out eggs. They're very highly anabolic, whereas the males really don't need to eat very much at all. They just need a bit of glucose to get around and get to the females. So I think that's why female guts deteriorate so much during aging and males don't. It's because of the very different digestive physiology, basically. Females have got bulk nutrients coming through. They've got to get it through that epithelium and into the fly, whereas males don't need to do that and can afford to elevate autophagy, which is probably quite powerful against infection in the gut. I think mice, it's much more complicated. Most of the time, male and female mice are rather similar to each other in physiology. Females do need to eat more than pregnancy, but the sex difference is nothing like as great as it is in flies. So I think that probably explains why flies are so dimorphic in the response to rapamycin and to trametinib, actually, whereas mice are not. But a lot of the same things happen. So I think female flies are basically a good model for mice, whereas male flies are not. So you see this deterioration in mouse guts of both sexes, and a lot of the similar things going on in response to the drug. As to what happens in humans, it's anybody's guess. I mean, I've looked into the possibility of looking at other phenotypes of patients who are taking rapamycin. But if you're dealing with an immunosuppressed transplant patient, it's very difficult to find an appropriate control, really, because there are so many other things going on. So I suspect that using prescription data for humans is actually not, it's going to require experimental medicine, the kind of clinical trial that Joan Manick's been doing, to really get at that. And we don't have very much data yet.
I was just curious about your thoughts on both of the drugs suppressing RNA polymerase 3, and if that's indicative of a slowing of metabolism. So, because they would be making RNA, ribosomal RNAs and tRNAs and such. I think
I don't understand the answer to that question. I think it's a really important question, and we need to look at it. And we're actually starting to do some direct intervention in individual tRNAs. But as you all know, Pol3 is responsible for the synthesis of a lot of short RNAs, so ribosomal tRNAs and some other short RNAs. And it's really not clear why suppressing its activity should be geroprotective. But it's a conserved effect. You see it in worms and in yeast as well as in flies. And I think we really need to understand far better what's going on. It's clear in the gut it's happening in the stem cells. But again, what we need is some cell biology to see why these cells are healthier as a result of doing that. So I think great question, and I don't know the answer.
Over here. Helena, thanks a lot for the fantastic talk. Quick question about the subnetworks that you were talking about, the co-occurrence of the hallmarks of aging predicting mortality. Is the message here that any of those four, when you have one of those, you start off a slippery slope and you develop disease and die? Or you think you need 3 or 4 of those simultaneously and only then you start entering some sort of a pathway to disease? And could you block one of them and basically prevent the set to become full? What do you think?
I think that's very interesting and different for different diseases. So if you look at those columns with the 9 hallmarks and the 83 diseases, it's really obvious that some diseases have got a contribution from all or a lot of them. Now, whether that's essential before you get the disease, I think isn't clear. But the association's there, whereas others, only one hallmark ever seems to play a role. It's really specific, the association. So I think it depends which disease.
Question back. So you're focusing, or you're mentioning in particular, tumorigenesis, activation of the immune response, and then in the fly, the hyperdivision, hyperplasia in the gut in the females. So these are all things that are focusing essentially on cell division, it sounds like. Is a major effect of rapamycin and trametinib simply damping down cell division, or alternatively working through checkpoint proteins to get rid of either improper or excessive cell division? Is that really what these all have in common?
I'm not sure they all have in common. It certainly might play a very important role in some tissues. But for instance, the reduced inflammation in the brain is almost certainly not a cell division phenotype at all. And interestingly, at the dose that we were using it, trametinib actually doesn't cross the blood-brain barrier. So it's not clear what the mechanism actually is there. It may be a peripheral mechanism that's then somehow being communicated to the brain. But certainly as far as the inflammation goes, that's not going to be a cell division phenotype there. On the other hand, in a highly proliferative tissue like the intestine, that may well be important. I think we really need to look hard at what's going on in these cells and what the actual mechanisms are. So
you don't think that the inflammation is via, for example, expansion of an immune cell population that then signals through the blood-brain barrier, for example?
It could be. It could be.
So you started your talk by talking about how, to this day, what is known to be one of the major enhancers of lifespan is exercise and diet, right? So would this combination of trametinib and rapamycin, it looked like a fantastic expansion of lifespan even in mice. Would that be even better than exercise now? Are we getting close to actually getting the pill, or do you think that would still be synergistic to exercise? Would exercise and diet still be doing other things?
I think anything I said in answer to that question would be speculation. I think at the moment it's clear that the diet and exercise interventions are the safe and effective ones. I mean, these potentially geroprotective drugs, they're a long way from appearing in the clinic. So the answer right now is lifestyle factors.
Can you hear me? Yes. It was a very great talk. I have a question regarding actually the treatment with rapamycin. Does it happen, or does it work when the organisms are still developing, or it can also actually work once the organisms are actually reach their adult stage? So where, for example, all this process of growth are kind of much more slowed down.
I don't know what the effect of either of these drugs would be if you fed them in the pre-adult period. So in the fly, we wait for the flies to come out as adults, so they have a completely normal development. There's a maturation period at the beginning of adulthood of the fat body, the ovaries and so on. So we let them go through that, and only after that do they get either drug. In the mice, we wait till they're a minimum of 3, sometimes 6 months old, which again is adulthood, they're reproductive. We never use the drugs during development.
That was an amazing talk, Linda. My question is sort of related to human data, I guess. So the proof lies in the pudding, and the pudding is, will these translate to humans? So we recently have started getting data around rapamycin, specifically in context of the Pearl trial, where there was the 6 milligram dose and the 10 milligram dose that was given to humans. It was compounded, but still. And the data has been mixed. We aren't really seeing much benefit. There are some benefit in terms of some weight loss, but not really any benefit in terms of clinical biomarkers. Of course, there's no measurement of lifespan or health span in particular, but the clinical biomarkers don't seem to be dramatically changing. What are your thoughts on that?
Interesting, but preliminary. And one thing I would say about rapamycin is that I would never envisage, if it ends up being used in humans, giving it chronically. It's so clearly, I haven't said anything about the memory effect of rapamycin in mice, but it's almost as big as it is in flies. We've got unpublished data on that now. So it's clear that you can dose very intermittently, but you probably do need to dose for long enough to get the dose high enough for the geroprotection. I think we probably failed to do that with our intermittent treatment in the mice. But I think the one thing that's come out clearly from the animal data, and that I would say is likely to be true in humans, is that the treatment should definitely be intermittent.
Thank you.
2 quick questions and quick answers.
So it's very interesting that the animals still die. And the question I have is, what is actually causing the death? And might that be another pathway that's worth looking at?
Yes, so there's a very important question. Are these drugs just reducing the incidence of cancers, or at least delaying the incidence of cancers? And also, is the cause of death different? I don't think for either drug the answer to that question is clear. They both do reduce the occurrence of tumours. So we've got both cross-sectional data, which is what clearly shows the delay. What's a lot less clear often is what the mouse died of, because you come in the next day and the mouse is dead, and already it's in a state where it's extremely difficult to say what it died of. It's not like postmortems in humans. You don't get anything like such clear information. So the cross-sectional data clearly show the delays. I think what we can't say is whether the array of causes of death is different in the controls and the treated animals.
Last question.
Thank you very much. is this difference in the intestinal wall for structure, is it the same in humans between females and males? And then the second question would be, given that these are FDA accepted drugs for different pathologies, is there, to your knowledge, is there any way to look back to see a survival of people on these drugs?
Okay, so two very interesting questions. I mean, there are intestinal differences between male and female humans. So, for instance, the array of gut cancers that occur in men and women during aging are different. You know, what the reason for that is not clear, but there are clear differences in incidence of different types of intestinal cancer. And there are also big alterations to the intestine, both in mice and in humans, during pregnancy. So, which of course is something that doesn't happen in males. And the aftermath of that may be part of the explanation. There's big expansion of the gut during pregnancy, particularly the small intestine. And, you know, that may well have sequelae or events that happen at the time which are just completely absent in males. As far as, you know, asking whether rapamycin does anything in the gut in females, basically, certainly in the UK, I don't know exactly what the FDA rules are, but if you wanted to do a clinical trial on that, although it's a licensed drug, you would have to go straight back to safety and efficacy for the particular regime that you're using. So you're not completely back to square one. A lot's been done, but you really have to go a long way back in the licensing process for the specific outcome and the specific regime that you're using in your clinical trial. And your outcome, at least in the UK, cannot be aging. It has to be something with an ICD code, which aging has not got. So actually getting where you want to get is a long and arduous process.
Okay, great. Thank you so much, Linda.